Hello and welcome to a fresh edition of WIP's Biochemistry, and this lecture will be discussing blood buffers or buffers in body fluids. Now, biological processes and molecular interactions taking place in our body are pH dependent. All cells in our body maintain a constant pH of around 7.0, and a small change in this pH produces a very large change in the rate of biological processes. Normal blood is around, the pH of normal blood, as we know, is around 7.4, and if it drops below 6.8 or goes above 7.8, it will lead to some sort of respiratory complications or cardiac malfunctions. The blood clotting process may change, and it can be fatal also. It can even lead to death. The change of pH can affect so many processes, so many biological processes. One of the best issues to understand is the functioning of enzymes and proteins. For example, the enzymes in our cells are very specific and function optimally at the right kind of temperature and right kind of pH. If the pH of blood goes out of range, the enzymes stop functioning, stop working. And sometimes the enzymes can even get permanently denatured, thus destroying their catalytic power, catalytic activity. This in turn, as you know, can affect lots of biological processes, lots of metabolic reactions in the human body, leading to various issues. The cells are mostly challenged by acids or H plus ions. Now here what we mean by H plus ion is a hydrogen atom from which an electron is lost, which is also referred to as a proton. Now sometimes in some equations H plus is also represented as hydronium ion, H3O plus, which is effectively just the same as H plus when we equalize or equate a metabolic reaction process. Now here I said cells are mostly challenged by acids or H plus ions. How does most of the H plus ions produced in the body? Here there are lots of reasons. Now one is obviously the acidic substances consumed in our food will add to the H plus ions produced in our body. In our body fluids, the food we have, if there are more acids, the acidity of the body fluids, including blood, will go up. Acidity will go up. That means pH will go down. Another contribution comes from H plus ions released during metabolic reactions. As a byproduct of metabolic reactions, H plus can be released. But the most significant amount of H plus comes from muscle action, skeletal muscle action. Muscles in the body need fuel to work, as we know, and this fuel is ATP. The hydrolysis of ATP to ADP lead to production of excess amount of H plus ions, and this will upset the normal pH, normal acidity of all the biological fluids in the body, including blood. Now additionally, we can consider the H plus ions produced from lactic acid also. Lactic acid, which is formed during vigorous exercise, is another reason for adding to acidity of blood. Now how body manages the pH, the rise or a fall in pH of body fluids. Here a constant pH is achieved primarily by biological buffers. As we know, buffers are mixtures of weak acids and their conjugate bases, like acetic acid, which is a weak acid, and sodium acetate, which is a conjugate base, is an example. There are other common examples also, like carbonic acid and bicarbonate ion. Now, biological buffers are immediate defense mechanisms against challenge by acids or bases. The theoretical definition for a buffer is an aqueous solution that resists changes in pH upon addition of small amounts of acids or small amounts of bases. So a buffer solution, when small amounts of acids or small amounts of bases are added, the buffer solution works so as to maintain a constant pH. We will see how it is done in biological fluids in a moment. The buffers are effective as long as the acid produced or the acid load is not very high and alkali reserve is not completely used up. Now let us see an example here of using 0.05 molar HCl. When 1 drop of the solution is added to 100 ml of distilled water, what will happen is that the pH will be dropped from 7.35 to 7.0. But in the case of blood, so as to drop pH from 7.35 to 7.0, we will have to add 25 ml of the same solution, 25 ml of the 0.05 molar HCl to 100 ml of blood. So that is the difference. So that is how the buffers combat with this hydrogen ions or acids or bases to maintain the constancy of pH in our body fluids, including blood. Now, see, the whole body contains around 5.5 liters of blood. So as to bring the pH from 7.35 to 7, the whole body will require 1,300 ml. That is 1.3 liters of the same solution. So this is how buffers becomes very effective in maintaining the constancy of pH in our blood. Now just think, if our body has got water instead of blood, what will happen? So that is how buffers achieve this. Now let us say what are the chief buffer systems working in our body fluids. Now the first one is bicarbonate buffer. This is the most important extracellular buffer, having highest buffering capacity. Another one is hemoglobin buffer. This is inside RBC. It's an intracellular buffer system of the blood. Then there is protein buffer system. And extracellular buffer systems together with the bicarbonate buffer will work. Then phosphate buffer system also is there. Now we will see all these buffer systems one by one, how they work so as to maintain the pH of body fluids.